So, hi, welcome back. Thank you, you two. This is the last session. We're talking about the CMTA's Patients as Partners in Research platform. You already know who I am, and I've just mentioned we've got Dr. Vera Friedman from the University of Colorado and Dr. Bipasha Merkinjay-Clavin, who's come all the way from Johns Hopkins University School of Medicine to be with us today. So, this is what we're going to be talking about. We're going to give a current overview of the Patients as Partners in Research platform. That falls to me to do that. And talk about some current opportunities that we have for you to get involved in CMT research. Also, this is why I've got these two lovely clinicians on stage with me, is to talk about their perspective on research studies and clinical trials, because we very much rely on their hospital sites to run these trials. And then we're also going to let you know how you can become a patient as partner in research today, because we need all of you for any of this research to happen. And then there will be some time for some Q&A. So this is our Patients as Partners in Research platform. I know it looks like a lot of numbers, but this platform is you. So the platform has grown so far to 6,617 profiles. That's all of you who've made those profiles for us, so thank you. It covers 49 different CMT subtypes within the platform, and so far we've supported CMT patients to get involved in over 27 different research studies or clinical trials. So this is the growth in numbers on the platform that we've seen just in the last few years. More and more of you are joining this platform every year. And every single time somebody fills in a profile, you don't see me, but I do a little happy dance. I'm the one who sees these coming in. So it's just really wonderful every time you take the chance to fill in a little survey for us and join that platform. What it has become is the largest global patient-reported data set for CMT. So it makes it a really powerful tool to help us understand CMT, what types of CMT you have. This is making a really significant impact on the advancement of CMT research through patient participation. All the research, clinical trials and studies you've heard about today can't happen unless you decide to get involved in them. And you can't do that unless you know about them, like you know that they exist. So this platform connects you with those opportunities. So it's only possible with your involvement. The more of you that complete that profile, the more powerful the platform becomes, the more research we can get done. So it's also a truly global participant base. I don't know if you can see the red dots on this map that just shows everybody all over the world. I like the ones in the middle, 22 people in Hawaii, thank you. This global base enables us to support patient involvement in studies with universities and pharma companies in different parts of the world. You've heard us talk about studies in Milan, London, Cyprus today. This is why it's useful that the CMTA is globally operating. We also share the analysed, these are the different studies and some of the different partners that we work with on those studies. So different universities, different biopharma and biotech companies, and we can only advance these studies. You've heard about many of those studies today. We also share this data, is what I was going to go on to say. We don't keep it to ourselves. Don't worry, we're very careful. We follow HIPAA. We don't reveal any patient details, but we can share the analysed data back with the research community. So we know what percentage of you have which different types of CMT. We know when you first got your symptoms or when you first got diagnosed. That's known as the diagnostic odyssey. We know how that's different for different types of CMT. So many questions that I can ask from this data, and I sit there happy with my spreadsheet. And this is presented at a global research conference in Copenhagen last year. So all of the scientists then know what we know. These are the current active Patients as Partners in Research opportunities. Please don't attempt to read them all. My aim of this slide was to show you just what the scale is here. And the ones at the top in bold, they're the ones that we've added since I last saw you at the last Patient and Research Summit last year. So it's active. We're adding new opportunities all the time. The one in orange, I know my colleague's going to tell you about. I'm not going to mention that one at all. But it just gives you an idea of the different types of studies, and they cover all of the different types of CMTs, including those with unknown types. So there's an opportunity for everybody to get involved. So without further ado, I'm going to move along to Vera. If you're happy to come up again, Vera, who's going to tell us a bit about how this works from her perspective and how you can get involved and what that looks like.
Hi, how's everyone's energy? We talked in our last session on demyelinating CMT about having realistic expectations for clinical trials and their design. And I also think we have to have really realistic expectations of our attention span in 2024 for people talking at you. So I'll try to be mindful of that. So I said this in the last session, and I'll just sort of say it again for the whole group. There is no such thing as clinical research without patients. Just doesn't exist. So the crux of clinical research is patients and everything that they contribute from the time that they come to clinic with something that none of us have seen before to the time when there are treatments that cure a disease, right? So I want to start out and talk about, the different aspects of how patients contribute. And so I'm going to start just by telling us a little bit about the Inherited Neuropathies Consortium again. This is led out of the University of Iowa by Mike Shai, and he has single-handedly, in what I think is a completely Herculean effort, connected 20 sites from around the world where there are people who have a specific and rigorous interest in CMT clinically on a research basis and sort of globally. And so the goal here was to say, can we be more systematic and can we study the natural history of CMT, find new genes, figure out how to measure disease and find treatments through connecting the great minds around the world together and getting patients involved from all these different sites. And there's various funding mechanisms, among them CMTA, that are listed here. So what are the goals of the CMTA as well as the INC in involving patients in clinical research? So the first is clinical trial readiness. So what does that look like? So we perform at these various centers around the world natural history studies in the different subtypes of CMT. So that means if you come to see one of us in clinic, you have the option through a protocol within the Inherited Neuropathies Consortium of being entered into a natural history database. And whether you have a known CMT gene that we're interested in studying over time or an unknown, both of those, we are collecting data to see how the disease changes over the course of, subsequent years. The other goal is to develop clinical outcome assessments. We talked about this already this morning. What are the best measures for a specific type of CMT that will get the drug, if it works, to show us that it's working? And then also biomarkers, right? So in addition to what we see at the bedside and to what you report to us, can we find plasma biomarkers, measurable objective markers such as MRI, to help guide these trials, make them quicker, more efficient, and more effective? The other thing that INC has taken really seriously, and I really want to emphasize this because I don't think we've talked about this a lot today, is in addition to, all the trial readiness we talked about, they're involved in training the next generation of investigators in order to retain talent and interest in CMT. And that's something that I've benefited from personally very tremendously in my own career. And I think is just something that, if missed, can lose a lot of opportunities to keep people thinking about the field. So globally, the Inherited Neuropathies Consortium wants to involve talent from the investigator side, participation from the patient side to broaden knowledge of CMT all around in clinic and on the research side. Okay, so the first issue that they're tackling, are we ready for trials in CMT, led to the development of the various outcomes that are now being used in the ACT CMT trial, as well as in a clinical trial, which you heard about today, SORD neuropathy. And so without patients, we wouldn't have any of what's on this slide. So patients are coming in and donating their time and their effort, and they travel from out of state sometimes once a year so that we can collect the CMT functional outcome measure or a pediatric measure or actually an infant measure in order to be trial ready. And it's an enormous contribution and commitment. And what you see in that blue arrow is that thanks to the efforts of Mike Shai and the many investigators throughout the INC, we now have really spanned the lifespan of people with CMT as far as being able to measure progression and have learned how these various instruments are performing in different subtypes of CMT. So I can't cover this in detail, but these are some of the publications that have come out of this effort. And so everything from rare to common, these are natural history studies published in recent years that use the instruments that were developed with patient participation to show progression in different subtypes with patient participation, right? So you're definitely seeing a theme here, okay? All right, and then what do we need to do as we're developing these instruments for established forms of CMT? We need to find new genes, right? And so I tell patients about this, and I try to really emphasize that it's almost science fiction, that in the last decade, thanks to Stefan Zuckner, who you guys heard from earlier today, and his collaboration with the Inherited Neuropathies Consortium, we've been able to pull so many patients from so many parts of the world into this one single database of genetic data that it's led to, genetic therapy discovery at an extremely efficient rate. And why is that important in addition to diagnosis? We're not living in an era anymore where we say, we have your diagnosis, you have CMT, there's really nothing else to talk about, right? And I think a lot of people in this room or their parents have heard that narrative, and it was real. That was kind of how the disease was approached for a long time. But of course, we're well beyond that. So these genes, as they're discovered, lead to new biology. The new biology leads to mechanisms that lend themselves to treatments. Treatments get developed, and that leads to clinical trials. And that's what we've seen. So I can't overstate this, but I don't think I need to. If you look on this slide, so in the past of this INC development and Stefan's work, there's been 30 new genes discovered in CMT. I would challenge you to find a rivaling situation in other related conditions. I mean, that's really astounding. And these are high-profile publications for a good reason. And so I'm not going to retell the SWORD story. I think you guys have probably been pretty saturated with it. But I want to tell you about it from my perspective. And so I think there's a lot of serendipity in this story as to why from gene discovery it took less than 18 months to go to a clinical trial, which is, again, sort of unheard of in biology. There was the accident of the mechanism of the disease being in an established pathway that we all already knew about. It's been looked at in diabetic neuropathy. There was the accident of a company having a targeted compound that, was directed exactly at this pathway that they had already spent a lot of time thinking about. So things came together. But what I want to say is that without the Inherited Neuropathy Consortium collecting data for years in patients who did not have a diagnosis, clinicians saying, I don't know what this is, I don't know what's causing this, but I think it might be genetic. Let's enter you into the inherited neuropathy. It's called the 6602 protocol, which is where we send blood to Stefan in Miami, and his group basically inputs the genetic data. So that was going on for many years before the discovery of SWORD. So first of all, it led to the discovery of SWORD because they had enough patients to see the pattern of what the gene was going to correlate to, right? But then once the gene was discovered, all they had to do was look back and say, okay, what do we know about the progression of this gene in these participants who've already dedicated their time and effort year in, year out? And that already paved the way for what you're seeing here, where we moved from, Applied Therapeutics, moved from in vitro to phase 3 in this extremely efficient way. And having participated in the trial over the last 2 years, I can tell you every time that I saw a participant back and we did all the various assessments, it is riding on the shoulders of all of the INC's efforts, that this was able to happen. And it's just enormously awesome, really. That's the word, awesome. Okay, so conclusions. So patient participation has been critical to the scientific advances we've seen in CMT from the very beginning. And I would say from the time of Charcot-Marie-Tooth. So not just when we discovered the first gene, but when clinicians around the world, looked at these patients and said, we're seeing a pattern. And there seems to be a certain, I come from a family of neurologists. And, I know that several generations ago, this was the conversation. It was very detailed clinical descriptions, and they were invaluable to everything that came later. So then, the second bullet on here is that we're not actually anywhere near done. So this in some ways is probably the most important time for patients to be involved as we're trying to bridge these discoveries and preclinical findings to clinical trials. And I think with clinical trial planning, at least in my opinion, the input from patients has to happen at every stage of this process. So it has to happen now before we even start planning our clinical trial. And we have to hear What do people think is important? What do they think is feasible? What are the parts of their disease that are most burdensome? This gets into all the patient-reported outcomes throughout the trial. The feedback from our participants drives a lot of what we do. And then everything up until the exit interview, where someone says, okay, what was your experience on this drug? You don't even know if you got the drug. You might have gotten placebo. Tell us, right? And all of that information is how we pivot, I think, towards making progress. And then the last issue, if we found a treatment for CMT1A, we heard an outstanding talk, today on a possible treatment that's coming to clinical trials. There's still a tremendous amount of work to be done, and there's a lot of genes that have to get discovered. And it's disheartening for patients who come to clinic with unknown forms that are progressing. And for that, we have to continue to enroll in this gene discovery pathway. And so I'll tell everyone here, if you have an undiscovered sort of unknown type of CMT, get connected with the CMT Center of Excellence that's within your driving distance, ideally, where you can come back once a year. I try very hard to connect the research visit with my clinical care of the patient. They're separate. Your research participation has no bearing on the care you get, but I try to make it logistically feasible. Like you come and see me in clinic, and then we kind of do the research piece, hopefully at the same time, to make it easier. So I think what we're going to see is continued discovery of genes and more patients involved as treatments become more feasible. And I'm hoping that, all of you will be part of that effort. And with that, just another thank you to the CMTA for their support, and a huge thank you to all of you for being here. And hopefully all of us will continue to work together.
All right. And so thanks so much for setting the stage, Vera, very nicely. I'm Bipasha Mukherjee-Clavin. I help co-direct the CMT Association Center of Excellence at Johns Hopkins University. And it's my pleasure to share with you today the story of the Johns Hopkins CMT2C, or TRPV4 neuromuscular disease natural history study, which is unique to Johns Hopkins right now. And I think that this study is a fantastic example of, first of all, patients partnering in research. And also, I think it's a nice example of how natural history studies pave the way for therapeutic interventional clinical trials. But first, I want to acknowledge the people who have done much of the work that I'm presenting today. So much of this work has been pioneered by Dr. Brett McCrae, who was at Johns Hopkins University up until recently, and he's now starting up his own CMT clinic at the University of Michigan. A lot of the infrastructure for this TRPV4 natural history study was put into place by Maya Hoke, and she's now a medical student at the University of Buffalo. And all of this work has been done under the leadership of Dr. Charlotte Sumner. She and I work together to direct the CMT clinic at Johns Hopkins, and she's been the world's leading investigator on TRPV4 neuromuscular disease for over the past decade. So I want to start by telling you a little bit about CMT2C, or TRPV4 neuromuscular disease more broadly, as we like to refer to it. And it's an unusual form of CMT. So these patients have a mutation in the TRPV4 gene that then results in a mutated TRPV4 protein. And that mutated TRPV4 protein then goes on to cause autosomal dominant disease that can cause muscle weakness in a few different patterns, and there may or may not be sensory loss. Examples of sort of 3 different patterns of disease that patients can have are the patient on the left, who we label as having CMT2C. This patient has significant weakness in their hands and feet, but they're relatively strong around their shoulders and hips. There are other patients with TRPV4 mutations who have significant weakness around their shoulders and their hip muscles, but they have relatively strong hands and feet, and those patients are labeled as having scapuloperoneal SMA. And there are also patients who have TRPV4 related weakness that's present, from birth or from infancy, and those patients we label as having congenital distal SMA. But what we're finding is that most patients with TRPV4 mutations don't necessarily neatly fall into one of these 3 categories, and rather we think that most of them exist on a spectrum. I think the really exciting thing about TRPV4 neuromuscular disease is that it's a potentially treatable condition. So Actio Biosciences in San Diego, California is currently developing a novel small molecule inhibitor that targets TRPV4 neuromuscular disease. They are finding that their inhibitor in their testing seems to target the disease-causing TRPV4 mutations with high potency, and they're currently testing their compound in TRPV4 mouse models. But the hope that we have at Johns Hopkins is that if everything goes well, the Actio Biosciences TRPV4 inhibitor compound can potentially be used in a clinical trial on TRPV4 patients to see if it helps improve outcomes in patients with TRPV4 neuromuscular disease. However, with where the field stands right now, it would be challenging to confidently design a therapeutic interventional clinical trial for TRPV4 neuromuscular disease. There are a number of different barriers, including the fact that it's a rare form of CMT and there's a very heterogeneous population. So patients can have things like scapular winging from the severe weakness around their shoulder muscles. They can have vocal cord dysfunction. They can have breathing dysfunction. They can also have bone development abnormalities. Another challenge with confidently designing a clinical trial right now is that the field lacks an understanding of how TRPV4 neuromuscular disease or CMT2C progresses over time. And the field also lacks a comprehensive understanding of the full spectrum of the types of symptoms that patients can develop. The field at present also currently lacks biomarkers of disease severity and progression that could be used to design that clinical trial. And so in order to address those barriers, at Johns Hopkins, we're currently performing the TRPV4 natural history study. So as part of this natural history study, patients with confirmed diagnoses of TRPV4 neuromuscular disease or CMT2C from all over the United States are invited to fly to Johns Hopkins once a year for 6 years. And basically every year they would undergo a 2-day battery of testing. And our goal enrollment is 60 patients. And so far, we're proud to have enrolled 12. You can also learn more about this particular study on the CMTA's Patients as Partners in Research website. So as part of this natural history study, during this intense 2-day battery of testing, patients undergo a number of different evaluations. They undergo a comprehensive clinical neuromuscular assessment. They undergo a vocal cord assessment by one of our ear, nose, and throat colleagues, a speech assessment by one of our speech and language pathology colleagues, an EMG nerve conduction study done by me, so I get to meet every single participant in this study. They undergo breathing assessments because that's an issue in this particular form of CMT. They undergo 2 different types of bone imaging tests to look for bony abnormalities that are specific to this type of CMT, as well as a special research protocol MRI to look for blood-brain barrier dysfunction that we think is also specific to this type of CMT. They also undergo a few different physical exam evaluations, including the CMT FOM, which has been mentioned a few times now this morning. So the CMT FOM is a clinical outcome assessment that's been developed by the Inherited Neuropathies Consortium, specifically to look for functional changes in patients with CMT, as Vera had mentioned. And it's also one of the outcome assessments that's being used in the SWORD study. So all of the work that we each do really builds off of each other. So I think that this work is incredibly important and potentially impactful. And the reason for that is that the natural history data that we collect from this TRPV4 natural history study is going to be used to design that therapeutic interventional clinical trial in collaboration with Actio Biosciences. And to me, this work is really exciting for 2 reasons. First of all, there's the potential that this compound that's being developed by Actio Biosciences could be one of the first disease-modifying therapies for a particular form of CMT. Second of all, I think that this natural history work is so important because there's going to be lessons learned from this natural history work and the later interventional clinical trial work that will be informative for clinical trial design for other forms of CMT. And with that, I'd like to thank you all for your attention. And of course, we're very grateful to all of the patients and families affected by TRPV4 neuromuscular disease. And we are actively recruiting, so feel free to reach out if you or anyone you know is interested in learning more.
Thank you so much, Vipasha and Vera. We do have representatives from Actio Biosciences. Are you still in the room? Waving. Hi. We held a focus group with Actio Biosciences last year. It was just one of the patients as partners in research opportunities. People came together over Zoom to talk about how the disease affects them, because the great companies working in CMT research seek to talk to the patients right from the very beginning to understand the disease, because there's no point in developing a treatment that fixes something that doesn't really matter to us. So they talk to us about what matters. And that's what Actio Biosciences have been doing, and also partnering with the clinical partners. And we do have time for probably one, maximum 2 questions. Did anybody have a question for any of us here? I'm going to quickly just pop this up whilst we answer those questions about how you can become a patient's as partner in research. Is a 22-question survey that you fill out on an online form, and you're in. And then we know about you. We can tell you about studies. And the QR code to access it is in your registration pack, where you can scan it now and you can sit and fill that form in whilst you're still in the room, and that'll have me do like 50 happy dances. So please fill the form in. I'm going to leave that up whilst I see if there's any questions.
I just want to say, Catherine, if we all fill that out now, will you do your happy dance on stage? Okay.
I will. Jill's going to do 5 minutes closing remarks, and I'll come back afterwards and do a happy dance. If Sarah Gentry tells me how many people have filled in a form, you can make it up, Sarah. It's fine. Any other questions that aren't about me doing a dance? Are there any questions on Zoom? No, thank you. We must have done a really good job. Well, I love to be ahead. 1 minute ahead. Please, may I therefore say thank you, because all of the studies you've heard about today are only possible because of you, the focus groups, the natural history studies, the discovering new types of CMT. Without you, we can't do this work. And I'm just ever so grateful for the clinicians who are driving it, and the patients who are taking part, and everybody who donates to fund it. It's a massive partnership effort. That's why we call it Patients as Partners.